Yes, morning everyone. And I think we are now 261, those who managed to log in. So I'm actually thinking that we should.
Morning, everyone.
I'm thinking that we should give some few minutes to the other students to be able to join before we start. Maybe we can give them like 5 minutes. Then we start. And again, you can also call your colleagues, share with them the link. Call them, share through WhatsApp, and remind them to join so that we can start. It is for all the students, not for specific groups. Everyone should be able to log in and attend the class online. Let me also share the class link, the YouTube link with the class rep. You can also post in the WhatsApp group. As we wait for your colleagues to join, let me mention that for these units, it is organized into 10 topics, which will be covered. Each topic will be covered on a weekly basis, and we have 7 lecturers who will be taking us through the various topics. So, and again, also to announce that the first cut will be done on week 6, that is on the 13th of October, while the second cut will be done on the 17th of November. Please note these dates, that the first cut will be done on week 6, that is 13th of October, while the second cut will be done on week 11, the 17th of November, and we expect all of you to be able to attend and do these cuts. Not unless perhaps you are bereaved or you are sick or you have another reason as to why you are not able to attend the cut. It will be a sit-in cut and not online cut. Please mark those dates because we expect at least all of you to be able to do those cuts. And I'm sure last week when you met the various lecturers in your groups, perhaps they may have mentioned that that the cut is always out of 30 marks, while the main exam is out of 70 marks. So the two cuts will give you an average of 30, and the main exam will give you 70. So you should not miss any cut, because if you miss one cut and you do the main cut and you do the main exam, then that will lead to that is what we call incomplete, and your marks may not be processed. So I want to encourage every one of you to be able to at least to purpose to do the two cuts and also to do the main exam. And again, all the classes will be done on Wednesdays from 7 to 10. So we'll have online class from 7 to 9. Then after that, we will proceed to meet our lecturers in the various lecture halls that we met last week. But again, if your class was on Monday, if you met your lecturer on Monday and not on Wednesday, then it means that you can only meet him next week. So those who are meeting lecturers today are only those who have their classes on Wednesdays. And let me just repeat that. If your classes were on Wednesdays, after this class, you proceed to meet your lecturer in the venue where you met last week. And the purpose is to seek for clarifications if there are issues that perhaps you are not able to understand in the class. And also the signing. You should be able to sign the class list, because that is also very key. So if you met your lecturer on Monday or Tuesday and not Wednesday, then you'll be able to meet them next week on Monday and Tuesday. That is for the face-to-face meetings. But for the online, it is for all the students, and the classes will be done from 7 to 9. Then after, we proceed to meet our lecturers. I hope that is clear. So I can see the numbers are still low, but I don't know, perhaps some of you are sharing the devices with other people. I don't know if that is the case, so that we can proceed. Are you sharing your devices with other people? Let me know through the YouTube chat, through the live chat in the YouTube, because I'm actually logged in the two systems. Let me know if you are sharing your devices with other people so that we can start. Because the number of people who are logged in the system may not be equal to the number that are listening. Is that the case? Yes, I think most of you are saying that is the case, that you are listening. You see maybe one device for multiple people, and therefore let's start so that we also don't waste time. So my name is Dr. Ndiwa. I'm from the Department of Biological Sciences, and my email is displayed on the screen. And again, I'm also the coordinator for this course, and if you have any issues, please let me know so that we can sort them as early as possible. And when you log in the e-learning, you'll be able also to see my contact as well as my email, because I can see that from the screen. It may not be that clear. So I'll be taking you through the first topic, which is I forgot to record. Please let me start recording. So I'll be taking you through the first topic, which is introduction to HIV/AIDS. And next week, you'll have someone else take you through another topic. The other week, you'll have someone else, and so on, until we cover the 10 topics. And so we can start. So like I said, the topic is just introduction to HIV/AIDS. And our learning outcome for this lecture is that we should be able to explain the meaning of HIV and AIDS. We should also be able to explain the various theories about the origin of HIV. What has people said about the origin of HIV? Because there are different theories which explain this. And again, we should also be able to explain or to give a synopsis of the history of HIV. I think that's not captured in the learning outcomes, but again, those are the three topics that we are going to cover this morning. So to begin with, what does HIV stand for? I know some of you are asking. The presentations will be from different lecturers on a weekly basis. But for me, I'm taking you through the first topic. So next week, you'll have someone else. The other week, there'll be someone else, and so on, until we cover the 10 topics which I have said. And all the notes are actually uploaded in the module. You should be able to visit the e-learning and download them. Please do not rely on your colleagues to get notes from the module. Just log in, because you have login details. Just log in and download the materials. And there are also e-activities. There are some e-activities, maybe links to some other resources that can also enhance your learning. So, so what does HIV stand for? I would understand by HIV. I think we met with some of you last week, and I know that you may have some prior knowledge on the same. So what does HIV stand for? I can't see answers in the live chat. Okay, so the HIV stands for human immunodeficiency virus, and this is the virus which causes HIV infection. So it stands for human immunodeficiency virus, which is the virus that causes HIV. And again, it is transmitted from one person, of course, through different routes. It can be transmitted from infected blood, semen, or vaginal secretions when they come into contact with either with the broken skin or mucous membranes that have ulcerations, what we call ulcerations. So it may enter through broken skin or through mucous membranes. And so basically what this means is that our skin and also the mucous membrane, they must be the first line of defense in preventing any disease-causing agents. And in situations where the skin is broken or the mucus has ulcerations, then that becomes obvious routes of entry of HIV and also other disease-causing agents. So the route of entry is basically through broken skin or mucous membranes, and this is through materials. When someone comes into contact with infected material such as blood, semen, or even vaginal secretions. So those are the primary sources of infection of HIV, because they tend to have a higher concentration of the virus. Let me mention something also about the mucous membrane. And the mucous membrane is actually It is a thin tissue which is found covering various internal organs. So it is found in the internal organs of various organs. You may find it in the gut, in the various parts of the gut, that's in the mouth, in the rectum. You may also find it, it is also found in the eyes, in the nose, and also in the various reproductive organs like the vagina and also the opening of the penis. So they kind of protect these internal organs from various infections. And therefore, like I said, in situations where someone has wounds or there are ulcerations in these mucous membranes, then HIV can easily enter into someone if semen or blood or vaginal secretions are contaminated. So again, also it also points out that sexual intercourse or sexual transmission is a primary route for transmission for this disease. I can see some of you, you are giving your definitions of HIV, which are correct from the live chat. So, and that's good. I can see Winifred Mutunga and the others. That's the correct definition of HIV. It stands for human immunodeficiency virus. And in addition also to transmission through contact with these infected materials, semen and vaginal secretions, HIV, it can also be transmitted from mother to child, either during delivery, during pregnancy, or during breastfeeding. And again, this also points out that HIV can also be excreted. It can be shed through milk, if that is one of the routes of transmission. Please also note that people with HIV, some of the people who are HIV infected may also develop what you call AIDS. And AIDS is actually the most advanced stage of HIV infection. But I'm saying some, not everyone. Not everyone who has HIV infection eventually develops AIDS. So it is just some of them and not everyone who is HIV infected, just some. Just a minute, I want to get some tools here. Okay, we can proceed. So those are some of the routes of HIV transmission. Of course, there will be a topic around this area. There will be a topic around HIV transmission, which will be done later. So I just pointed out a few routes of transmission. And let's say also look at a few features of this virus. So there are two major variants of HIV, what we commonly refer as genotypes. But now, because I don't want to complicate this lecture, we can refer to them as like different types of HIV viruses. And here we have two major ones. We have HIV-1, which is very common, which is the most common type of HIV virus, and which actually accounts for majority of HIV infections globally. So it accounts for about 98% of all HIV infections worldwide. Very prevalent, very common in terms of geographical distribution and in terms of prevalence, how many people get infected by this particular type of HIV virus. And again, also compared to HIV-2, it is also more lethal. It is very virulent, so it is more lethal. And again, it also progresses more faster compared to HIV-2. So HIV-1 is actually very common. It is more pathogenic, it is more lethal, and it accounts for majority of HIV infections globally. And it is about 98% of all HIV infections. Now, the second type or variant of HIV is HIV type 2, HIV 2, and which again it is less prevalent. It is not as common as HIV 1, and it is very much restricted to the Western Africa, where it affects majority of people. So you can actually say that HIV 2 has a limited geographical distribution compared to HIV 1, which will be found in all the continents. HIV 2 is mainly restricted in West Africa, and again, it is not as pathogenic, it is not virulent or fatal compared to HIV 1, and it also progresses slowly compared to HIV 1. I can see some of you from the live chat, you are saying that the notes are not clear. Perhaps, Serem, is there a way we can improve the clarity of the notes so that they are clear to the students? Mr. Serem, I can see you are logged in. Is there a way we can improve the clarity? Because from the YouTube, the slides are not that clear. Perhaps maybe doing some adjustments, I don't know. Mr. Serem, there are complaints about clarity of the notes in the YouTube, and even from my side, I can see that they are not clear. I'm trying to, please let me also try to conduct Serem so that the notes are clear from the slides, so that you can follow what I'm saying. Just a minute, then we proceed. Say that the notes are okay. So what can they do? And but some of you are saying that the notes are clear. So I don't know which is which. Most of you are saying that the notes are clear. Some of you are saying that they're not that clear. Yeah, they are clear. So I think we just proceed, because majority of you are saying that are clear. Perhaps you can just log out and log in. I don't know, but he say, let me see what Mr. Serem is saying about this. That the notes are okay. Only they need to use the high resolution video. So the solution is to use high resolution video. I don't know how that works for improved clarity. Okay, so let's proceed. Let me also log into the YouTube again. So I was saying that we have those two types of HIV virus, and they differ in terms of geographical distribution and also in terms of virulence, and how they progress after initial infection. And also, even within these two types, you can also find subtypes. There are also strains, as well as what we call recombinant forms. And these subtypes, or the strains, they can be either within the two major groupings. And again, you can also have a recombination between one strain and another one. And again, this is also because of the fact that HIV is a virus which is highly mutating. It mutates, it has a very high mutation rate, and that leads to various strains. Again, also knowing this, it also emphasizes that that people should take precautions, especially if they are HIV infected, because there's that high likelihood that individuals can have either co-infections by different strains, which can lead to, of course, to various health outcomes which may not be desirable. So very important that people be careful if they are HIV infected, because they can have two or multiple infections with the different strains, and that can lead to various health outcomes which may not be that good. It can worsen the health condition of the person who is infected. And so let me move to the next slide. So again, also on the same about HIV virus, it is a 20-sided virus. So we talk of icosahedral, meaning that it is a 20-sided virus, which also has an envelope. So it is surrounded by an envelope, and it is also an RNA virus. Last week when I met my group, we did a short discussion about HIV, and I learned that you are familiar with things like RNA. Some of you, of course, it may not be everyone, but majority of you are familiar with what is RNA and what is DNA. RNA stands for ribonucleic acid, while DNA stands for deoxyribonucleic acid. And these are genetic materials. So different organisms may have different genetic materials. And in the case of HIV, the genetic material is RNA, ribonucleic acid, and not DNA, like in the case of humans. For us, we have DNA. Of course, now the DNA in our bodies leads to formation of RNAs and so on. But I really don't want to go that route. Just want to emphasize that HIV is a RNA virus, which belongs to the family lentiviruses, subfamily retroviruses. So what are retroviruses then? What are retroviruses? And how are they unique from other different types of viruses? Is there anyone who has an idea of what are retroviruses? Teresia Kasamba, what is the meaning of the word mutation? Please ask as many questions as possible, things that may not be clear. Mutations means that the genetic material of that virus keeps on changing every time as the virus is multiplying. So it changes. It changes, and that now creates a problem. So you end up creating, like, you can think of, because mutations leads to different strains, like I said, you can think of these different strains as different viruses like that. But of course, it is as a result of multiplication of viruses, and which leads to changes in the genetic makeup of that virus. So let me explain what retroviruses mean. And on this, I will not give much details because they may not be necessary. But retroviruses are very unique type of viruses which are able to make DNA from their RNA. Remember, I said that HIV is a RNA virus. So after entering into the cell of the host, let's say into the host cell, these viruses are able to make DNA from their RNA. So you can think of them like they tend to hijack the replication, the DNA multiplication mechanism of the host. So they put their DNA inside the cell, and then as the cell is multiplying, the RNA of that virus is also multiplying. Because retroviruses, because when you look at the way genetic material flows, like in our case, like I say that we have DNA, right? And then the DNA will make RNA, and the RNA will lead to the formation of proteins. DNA to RNA into proteins. That's how we expect genetic information to flow, right? However, in the case of retroviruses, they do it in the reverse way, from RNA to DNA. So that's why they are called retroviruses, because they work in the opposite of what we know is the way the genetic material is supposed to proceed in terms of how information flows. Please just give me 1 minute. 1 minute. I plug my laptop to power. Just 1 minute. So that's how we expect the genetic material to flow in a normal circumstances, from DNA to RNA and then into proteins. But now that is complex, which I really don't want to go into that. Just to emphasize that retroviruses do not use that normal way we know the information is supposed to flow, but rather they make RNA from DNA. So they work in the opposite way. That's why we are talking of retroviruses, retrogressive way of working. Like they are working backwards from the norm of how we know information flows. And in addition to this, we also have another type of virus, which is called simian immunodeficiency virus, SIV, which is also very close to HIV. And both are also retroviruses. They also belong into the same family of lentiviruses. But again, simian immunodeficiency viruses, SIV, are mainly infect monkeys and also non-human primates. These are the likes of chimpanzees and also gorillas. So those are the primary hosts which are affected by SIV. But the SIV is very close. It is, in fact, it is thought to be descendant of HIV. Sorry, HIV is thought to be descendant of HIV because there is a lot of similarity in terms of how these two viruses looks like. So it's a very close virus which also affects non-human primates, the likes of monkeys and also chimpanzees and also gorillas. And we also have other different types of viruses which are also similar to HIV. It's also one which is also called feline immunodeficiency virus, which tends to affect the felines, the cats, the cat family, and so on. But for us, let's just focus on these two, HIV, and also look at simian. SIV is another virus which also has a similar, it also causes similar symptoms in animals and not in humans. Although humans can also be infected, but again, they may not display severe form of the infection like the non-human primates. I hope that is clear. This figure is not meant to scare you. Just wanted to show you how HIV virus looks like in terms of morphology. And I say that it is an RNA virus, so that's the genetic material. And it is also enveloped, so it has this capsid, what we call capsid, or a cover, which surrounds the virus particle. And again, on the surfaces, it also has these knob-like structures, which it uses to attach and also to enter into the host cell. That's basically what I wanted you to learn from this figure, just to understand how it looks like, material, and also to emphasize that it is enveloped. It is covered by a capsid, and on the surfaces, it has these knob-like structures, which helps it to be able to enter into host cells. And I said that it makes DNA from RNA, right? Because of the presence of this enzyme, which is called reverse transcriptase. It is the one which helps the virus to be able to make DNA from RNA. That reverse mechanism which I was explaining in terms of how genetic material flows. Just to sample some of your comments, you are saying that the slide is not clear. So there is the recommendation was you use high resolution video. That's what we had from the ICT team. Perhaps you may just log in and you may log out and log in. I hope that works. Some of you are admiring the shape of the HIV virus. So that's basically the shape of HIV virus. And please note that the purpose was just for you to appreciate the shape, not for you to start memorizing what does GP stand for and so on. Just to know how it looks like, so that when we say that HIV is RNA virus, at least you understand what we mean by that, and you can know why it is a retrovirus. It's because of the presence of that reverse transcriptase, which I said really helps it to be able to make DNA from RNA. Okay, so what does AIDS stand for? What does AIDS stand for? How do you understand by these initials? How do you define? What does AIDS stand for? Yeah, I can see some of your answers from the chat. Gabi, you are saying that it stands for acquired immunodeficiency syndrome. Yeah, that's correct. I think, and also Mwiti, you also have the same answer, Amuka and the others. So that's actually the definition of AIDS. So the initials AIDS stands for acquired immunodeficiency syndrome. So again, let's look at briefly what does acquired mean, what does immunodeficiency mean, and what does syndrome mean. So for AIDS, the A stands for acquired, the I stands for immuno, and the D stands for deficiency, and S stands for syndrome. So the full word is acquired immunodeficiency syndrome. And for acquired, in this case, it means that the disease is not hereditary, right? And therefore, it develops after birth through contact, that's after contact with HIV in either semen, in either vaginal secretions, in blood, and so on. So it is acquired. It is not something that person inherits. And immunodeficiency, on the other hand, here, it means that the disease causes weakening of the immune system. There's weakening of the immune system, and this is because of the fact that the virus tends to destroy several types of white blood cells, specifically what we call the T helper cells. I think that will be covered sometimes later. There will be a topic on immune system and how HIV affects that. So the virus usually causes destruction of certain types of white blood cells, what you call the T cells. Of course, that will be covered later. And that destruction usually is the one which makes individuals more vulnerable to other disease-causing agents, because they are the soldiers. You can think of those cells which fight disease or infection-causing pathogens as your soldiers. And if they are destroyed, then individuals then become more susceptible to other disease-causing agents, which healthy people are able to fight without any problem. So that's one. So there's weakening of the immune system, and individuals become vulnerable to other disease-causing agents. And the word syndrome, in this case, it refers to the symptoms that collectively indicate or characterize a disease. There are many symptoms, of course, that characterize AIDS, because like I said, AIDS is the advanced stage of HIV, right? And individuals may manifest, different types of cancers that will be covered later. They may also have a diminished number of various soldiers, what I call soldiers, the cells that fight infections, what we refer to as the CD4 cells or T helper lymphocytes, to be very specific. They are the soldiers, the cells, the white blood cells, which play very major role in fighting infections. And because of the invasion of this virus in these cells, then they are destroyed, and that will lead to decline in the number of the T helper cells or the CD4 cells, which I said is the major cause of weakening of the immune system, and of course making individuals to be more vulnerable to other diseases. And these are very important cells. in our immune system, because actually without this, then individuals may not have a well-functioning immune system. Because even apart from fighting HIV and other infections, they also work with other cells. They also activate other cells of the immune system so that someone has a better response to infections and so on. So I don't want to overemphasize on this, because there will be a topic covering on AIDS, some of the symptoms that are manifested by individuals who are infected, and of course examples of these examples of some of these other opportunistic infections which may also infect individuals, like different types of yeast infections and so on and so on. Just to mention that the symptoms of HIV are many, and that will be covered in more details later. So what does CD4 stands for? What type of cells are they? CD4, the word CD stands for cluster differentiation, and this is very complex. I really don't want to go into more details, but it stands to cluster differentiation. And basically, these are proteins which are found on the surfaces of the cells, right? And so such type of proteins are used to classify different types of cells. We have CD4, it's also CD8, CD16, and so on. So they're different. And cells which have these protein membranes on their surfaces are basically the T helper cells, the T helper lymphocytes, type of white blood cells, or simply T helper cells. They're the ones which have these protein membrane on their surfaces. Very typical of this type of cells. I hope that is clear. Yes, CD4 are white blood cells from Mutinda and the others. They are white. They are type of white blood cells. We have many different types of white blood cells. So this is just one type of those leukocytes which help in fighting infections. So how does then HIV cause AIDS? How does HIV cause AIDS? How does it cause AIDS? Saitoti, Lusuna, you are saying white cells. Not clear what exactly you mean. I can't see your responses from the live chat. So what does HIV, how does HIV cause AIDS? Okay, let me explain this, but already I have explained this, that HIV usually attacks and they destroy certain types of white blood cells, the CD4 or the T helper cells, or basically T helper lymphocytes. And I mentioned that they are very important in our immune system. For a proper functioning of immune system, you need CD4 cells. And they are also very important in fighting infections. So they attack, the virus attacks and destroys these cells. And in fact, when this happens, when the CD4 cells are destroyed, then there's high likelihood that the individual will develop AIDS. Of course, some people who are AIDS infected, they might take long. On average, this could be like 10 years, 10 to 12 years, if the individual is not under any ARV and the retroviral treatment. If they're not under any treatment, HIV individuals may take like 10 years before they develop AIDS. But of course, this could be variable depending on the individual, and of course other factors like the person's health status, for instance, can influence the development of AIDS. Health issues, diet health lifestyles, some behaviors like intake of alcohol, they can also increase the progression from HIV infection to AIDS. And again, so the key message from my slide, from this slide, is that HIV causes destruction of these soldiers, the CD4 cells, and that is the one which leads to, later it leads to HIV because of what I said, a weakened immune system. And again, like I said before, not everyone who has HIV develops AIDS. That's also something that you should also be able to consider, because AIDS is the advanced stage of HIV infection. And for people who are HIV infected, but they are not under ARVs, they may take on average about 10 to 12 years before there is enough damage of their immune system for the AIDS to be able to develop. And lastly on this is that, you know, the development, the progression rate from HIV infections to AIDS is very variable depending on individuals, and this could be affected by other factors like the health status of the person, you know, things like diet, health, other factors, you know, like some behaviors like, you know, intake of alcohol and so on. They play very major role in the progression of HIV infection to AIDS. We'll be able to cover this again later. We'll look at the relationship between, for example, alcohol and HIV progression, as well as look at relationship between HIV and use of other drugs like intravenous drugs like heroin, cocaine, and so on. So that again will be covered later at some point. Ezron Cheme, you are saying that you not understand very well on how HIV causes AIDS. I think I've repeated that. But I said that it basically destroys the CD4 cells, weakening the immune system and making the individual more susceptible to other disease-causing agents. That's basically what I said. Destruction of your soldiers, the soldiers that fight infections. In this case, the CD4 or the T helper cells, which are very key in fighting infections and also activating other cells of the immune system to function effectively. I hope now that is clear. Cynthia Chipkrui, cluster and differentiation. I said you can think of CD, the cluster differentiation is proteins which are found on the surfaces of the cell. They're just proteins, and I said we use them to be able to classify different cells of the immune system. So you classify them based on what type of protein is found on the surfaces of their plasma cells. And I said on these that for T cells, they are the cells that have CD4. We still have other types of CD. We have CD8, will be found in other type of cells, and so many, which I said that I really don't want to go into discussing these different types of glycoproteins or proteins which are found of the surfaces of the different cells of the immune system. And because you are not doing biology, of course, so let me just stop at that point in terms of what we know about the T cells, what they have on the plasma, on their cell membranes, because that is enough for this unit. And so now, having looked at the various definitions of what is HIV, what is AIDS, and of course try to relate that with SIV, the virus which I mentioned affects non-primates. Let's look at some historical perspective and overview of HIV. When was the first case, for example, reported, which year, and also look at some of the milestones in terms of what has been done in the management of HIV That's in the management of HIV, and also measures that perhaps have also been taken to prevent transmission. And just to note that before we look at this is that even though the first known case of HIV in humans was reported in 1959, HIV may have been in circulation or in existence as early in 1920s. That's based on some literature. It may have been in circulation as early in 1920s, but perhaps in low circulation, maybe the number of people who were infected at that point may not have been that many. And so you need to note that before it was detected, there are people who think that it may have been in circulation as early in 1920s, in 1930s, and so on. But the first known case of HIV was actually from a blood sample collected in 1959 from a man in DRC, Congo. But again, how this person became infected, we don't know. So we have very limited data. We don't have a lot of information with regard to how it became infected with HIV. Also to note that it was up to 1980s. So up to 1980s, again, we also don't know how many people were infected with HIV or the percentage of people who were infected with HIV and developed AIDS. So again, that information may not be available because a lot of information about HIV came from, it was available from 1980s and onwards. But however, also to note that even at 1980s, some literature also suggests that HIV may have spread to several continents by this time. So it was actually spreading to all the different continents in around 1980s. But before that, and before 1980s, we really don't know much about this virus, although it is thought that the virus was still in circulation in human population. And again, also to extend on the same, is that in the case of USA, there is evidence that HIV was in circulation. There's some information that HIV was in circulation in early mid or even in the late 1970s. And actually, between the year 1979 and 1981, doctors in USA, in various hospitals, were actually reporting some rare types of conditions which are particularly associated with HIV. And this includes pneumonia, rare types of pneumonia, to be specific, pneumocystis carinii pneumonia, PCP, very rare type of pneumonia, and also other types of cancers like Kaposi sarcoma, which are typical, which are mainly found among HIV AIDS patients. And of course, these individuals also had what we call opportunistic infections. These are diseases which do not actually affect healthy individuals. People who have a healthy functioning immune system may not be able to get these opportunistic infections. But they were very common among male who are having sex with other men. So again, also to note that this information about these symptoms of HIV were being reported among the gay community, people who were having sex with other men. And it was actually up to 1981 that HIV was first described. Again, also this is based on this information that was being gathered by these doctors in USA, to be specific in California and so on. And then what happened next in 1982 is that the CDC, what we refer to as the Center for Disease Control, they began to use the term acquired immunodeficiency syndrome, that's AIDS. And again, in some parts of the world, like in the USA, they also began formal tracking of HIV/AIDS cases. That's formal tracking basically means that looking at surveillance data, how many people are getting infected with HIV/AIDS, and how many people are progressing from HIV infection to AIDS, and so on. And I think some of you are saying that yeah, I'll send you these slides. I'll send you these slides. They are kind of enhanced with some new ideas, with some new notes. So I'll send you after the class. And again, the link for this class will also be posted in the module, so that if you have missed anything, you can also listen to the presentation. But the link will be posted tomorrow, not today. It usually takes some time to get the link to the presentation. So I was saying that in 1982, that's the time when some of the countries like USA began this formal tracking or surveillance to determine how many people were getting infected with HIV virus and how many were progressing to get AIDS. Then come 1983, this is the time when HIV virus was isolated by some scientists in France, and they named the virus lymphadenopathy associated virus. So the virus was first isolated in France. That was in 1983, and the scientists there, and later the virus was named lymphadenopathy associated virus. You may have seen this when you are reading through the various materials. You may have seen that HIV was known by different names before it was later named HIV by the International Scientific Committee. So the first naming was actually done by this International Scientific Committee, which deals with what you call taxonomy of viruses, the naming of viruses. That is the mandate. And again, also in the same year, the Center for Disease Control, CDC, also identified the major routes of HIV transmission, but they also ruled out that HIV cannot be transmitted either through casual contact. It cannot be transmitted through water, food, air, or on surfaces. So this is also the same time when the many routes of transmission of HIV were also mapped out. And again, also CDC also came up with some recommendations for healthcare workers and other people who are involved in the health sector. So they came up with recommendations that were to be followed to ensure that HIV transmission is prevented. And also during the same time, the World Health Organization also had their first meeting, and that's to assess the global situation of AIDS, and also they began international surveillance. What I mean by surveillance is basically following up on how many cases are being reported on HIV/AIDS, how many people progressing to AIDS, and so on. Basically trying to get a lot of data about this pandemic, which was spreading in various continents. Then come 1984, again, another lab based in USA also isolated the same virus. And this time they referred to AIDS as human T-cell lymphotrophic virus type 3. Human T-cell lymphotrophic virus type 3, HTLV3. And so so when you look at the first case, I mentioned about a different name, right? I said that the lab in France named this virus as lymphadenopathy associated virus, LAV. However, a year later, in 1984, another lab based in USA also isolated the same virus and they came up with a different name. And this time they refer to it as human T-cell lymphotropic virus type 3. And again, also in the same year, in 1983, also the first case of HIV was also described in Kenya in 1983. Then the following year, in 1985, the USA Food and Drug Administration, FDA, also licensed a testing, an antibody testing kit, testing a kit, which was to be used to screen individuals for antibodies produced against this virus, and also to screen blood donors before transmission. So there's a lot of progress in terms of what we knew from 1980s. Every year, new things were being done, of course, to ensure that HIV transmission is being to ensure that there is no HIV transmission, or to reduce the number of people who are getting HIV. And these are actually in 1985. This is actually a significant step, because with the proper testing, then basically that means things like blood donors, use of blood for transfusion, it could not lead to the transmission of the virus, because you are able to detect the antibodies against the virus at that point. And again, in 1986, now this is the time when the two names for this virus were harmonized. So the two names which I said actually were abandoned, and now a new name called HIV, that's human immunodeficiency, was adopted by the International Committee of Taxonomy of Viruses. So the two other names were actually abandoned, and from that point, now we had a new name which was to be used to refer to these virus which causes HIV. And again, also in the same year, HIV-2 was also identified. This is the virus which I mentioned in my initial slides. It is very much common in the Western Africa, and usually it is not as virulent as HIV-1. So again, it is also during this same time in 1986 that HIV-2 was actually identified as another variant of the HIV virus, which also causes AIDS. 1987, again, also there were also some other implementations done by the WHO, that's the World Health Organization. They also came up with a program on HIV/AIDS, just to raise awareness and also to provide technical as well as financial support to countries to be able to deal with the HIV. And again, this also led to encouraging participation of NGOs, as well as promoting the rights of people who are infected with HIV. It is also in the same year that also the first antiretroviral drug for treatment of HIV was actually approved by the FDA, the USA Food and Drug Administration. And this drug is called AZT, or simply Zidofudin. It's the first drug that was approved by the FDA for the treatment of HIV in 1987. Again, also in the same year, we also have, there's also confirmation by the World Health Organization that HIV indeed could be passed from mother to child through breastfeeding. And also during the same year, in 1987, the World Health Organization estimated that about 5 to 10 million people were actually living with HIV globally. And I'll just summarize just a few things. I really don't want to take you through all this, just to point out a few things of what happened up to the current situation that we have. So, and that's why you can see some of the years, there's no information in what happened in some of the years. So in 1988, again, WHO also declared that 1st of December was the first World AIDS Day. Then come in 1992, AIDS was actually identified as the leading cause of death among people aged between 25 and 45 years in the USA. Then I'll jump some few years. Now in 1995, again, another drug was also approved by the FDA. And now this ushered an era of a combination therapy, what we call antiretroviral treatment, the highly active antiretroviral treatment, where three or more than 3 drugs are used to manage HIV. So this is also another significant milestone in terms of management of HIV, because you can imagine that as new drugs become available, then basically that means there will be better management of the pandemic. So in fact, even when you look at literature, it shows that during the same time, countries that were able to afford these, in countries that were able to afford these drugs, there was significant decline in terms of people who were progressing from HIV infection to AIDS. And that led to a decline of about 60% of the people, again, who are also dying because of HIV/AIDS, as well as people who are being hospitalized because of HIV. It declined through this combination therapy, the use of multiple drugs to manage the HIV infection. And now this is now the last slide. I really don't want to bore you with a lot of history. So this is the last slide on the history. And now come in 1996, again, another drug which is called nevirapine was also approved by the FDA as a drug for treatment of HIV. Then in 1999, WHO, that's the World Health Organization, announced that AIDS was the 4th biggest cause of death globally. And of course, it was #1 killer in Africa. So this is the time when there were many cases of HIV and mortalities in most countries in Africa, including Kenya. And again, also in the same year, remember I said that in around 1980s, the number of people who were estimated to have contracted HIV were between 5 to 10 million, right? Now in 1999, this number had increased, and the number was actually about 33 million of people who were actually thought to have been infected with, were estimated to have been infected with HIV, and about 14 million of these people died from AIDS since the start of that pandemic. And then the last 2 milestones is that in 2006, again, male circumcision was also identified as one of the factors which could reduce HIV transmission from female to males, which could reduce the risk of HIV transmission of female to male, and that is in about 60%. The most recent milestones is that in 2012, again, the FDA also approved a pre-exposure prophylaxis. These are drugs which are used by negative people, HIV negative people, to be specific, to prevent the sexual transmission of HIV. But again, these drugs, they are also used in cases of rape. If someone is suspected to have been raped by someone with HIV, they are also put under this PrEP to ensure that they don't contract HIV. And then lastly on this is in 2017, it was actually estimated that people who are HIV positive That's more than 50% were actually under ARVs, and that's about 19.5% million people. So the purpose of this history about HIV is not for you to start memorizing what happens year after year, but basically for you to be able to appreciate what we knew about HIV in 1980s and what has been done so far. You know, the various milestones that that we have in terms of the information that we have about HIV, how new drugs were developed, and how the world is managing HIV pandemic. So that is the primary objective of this history section about the HIV. Just to have that knowledge to understand, not for you to start memorizing what happened a year after a year. And some of you are asking, let's be informed whether this history would be tested. Already I mentioned something about that. And how does circumcision reduce the rate of HIV transmission? Basically, this is by reducing the surface area that is in contact with vaginal secretions and so on. So that is the whole point of circumcision, basically to reduce the surface area. You know, when that part is cut, so the surface area will be small, and the surface area would be small that is in contact with potentially infected HIV secretions, like vaginal secretions, and also and so on. Respond to our questions, please. I was not able to look at all the questions. There are many. You are 384, and the comments are coming thick and fast. So I may not be able to answer all the questions, but maybe at the end I'll look at them and see what we can clarify. And again, also the fact that you'll be able to meet your lecturer after this lecture, you can also seek clarifications from them for the questions which I may not be able to answer. Where do we get FDA? This is it is in USA, on an organization which deals with approving of new drugs once they are developed. Date of the Cut 2, please check in the course outline. The course outline usually has all the details on when the first cut will be done and when the second cut will be done, as well as the dates for the main exam. And also information on what will be covered on a weekly basis. Some of your questions can be answered if you make the e-learning model your friend. You'll be able to see all this information. Now, so I want us now to look at something else, the last part of this lecture, and that's on common theories of origin of HIV. What has been put forward to explain the origin of HIV? What we know, where and when was the HIV first discovered, you know, and you know, to understand what are some of these hypotheses that have been proposed to explain the origin of HIV. I want to mention that when and where the HIV virus first emerged has remained unknown for many years. So there's a lot of controversy. There's no clear information on this. But like I said, that there are many theories that have been put forward to explain the origin of HIV. Of course, some of them are not that convincing. They have their own weaknesses. And I would like to invite you also to interrogate them, ask questions, and find out if you find them convincing, and so on. And again, also to mention that there is no consensus among people on these theories, but people may think otherwise about them. So let's look at them quickly, the common ones. And the first one is religious theories. Is HIV as a result of God's wrath, or is it as a result of some supernatural powers like witchcraft? No, that's what we seek to understand. So there are many religious groups that actually believe HIV came from an angry God, who was unhappy with some practices like homosexuality and also promiscuity, since these are some of the vices which are actually condemned by the scriptures. So there's that section of people who think that HIV is as a result of punishment or a visitation by God because of increasing, homosexuality in some parts of the world, or because of increasing promiscuous behaviors among the people. So there's that section of people who believe that it is as a result of God's wrath because of these vices that are increasing in our modern society. And we also have many Africans who also believe that HIV is as a result of witchcraft. And they also tend to use anti-witchcraft rituals to counteract the infection. So these are highly entrenched in our communities, these religious beliefs, and also these supernatural powers, which many people or some people may think that they are the major causes of HIV. And yesterday I was reading about the role of witchcraft and basically looking at HIV and witchcraft in Africa. And not that I like witchcraft, perhaps I have to be very categorical on that. And I came across one of the articles which is quite interesting, that perhaps you may also need to look at it at some point. The paper done by someone called Adam Ashforth. It is a study that was done in South Africa in 2002, and they're looking at relationship of witchcraft in the community. Very interesting perspective of how the community in South Africa looks at AIDS as a result of witchcraft. So I want to invite you, when you are free, look at this and you'll be able to understand what people think about HIV as a result of witchcraft. Of course, some of these beliefs are not true, they are unfounded, and we just need to demystify them. But HIV is not as a result of witchcraft. There is enough evidence, enough data to show that HIV AIDS is caused by a virus, like I mentioned. That virus is HIV virus. So these, some of these beliefs do not hold true in regard to what we know about HIV and transmission. The second theory is the contaminated needle theory, or what we refer to as the theory of serial passage. And I want to mention that in 1950s, this is the era when the use of disposable plastic syringes was actually it became very common in 1950s, because they were cheap, and of course it was easy they were used to administer medicines. But however, during the same time in Africa, here we are having challenges of populations. There's a huge population of Africans. So that means again also a huge number of these syringes would be required, which of course would lead to very high costs. And because of that, it is thought that some of the African healthcare professionals were actually vaccinating people using that is a single syringe among the in the Africa could be used to vaccinate multiple patients without disinfection in between. So because of that, it is thought that perhaps that practice of reuse of needles and the syringes could have contributed to the transmission of HIV from one person to another. So basically, that's what this theory is about: serial transmission through reuse and through the reuse of syringes and needles. You use one person, you also use the same syringe to another person, and so on. So that leads to serial transmission of HIV from one person to another. So it's a theory which again has also been put forward by some people to explain the origin of HIV and so on. Some of them are very weak, and that's why I'm inviting you to actually to interrogate all these theories and try to look at what are the weaknesses, what are the strengths, do they hold true? Do they hold true in terms of what we know about HIV transmission? Then again, also there's another theory which is called the under theory, and just to mention that diseases which are transmitted from animals to humans, we refer to them as zoonosis, right? Diseases that are transmitted from animals to humans, we basically refer to them as zoonosis. And HIV is an example. It is thought to be one of them. Of course, we think that it came from animals into humans. It's some evidence towards that. So we can actually refer HIV virus as an example of these zoonotic diseases. And again, it is also widely accepted that HIV is a descendant of the simian immunodeficiency virus, what I mentioned as the SIV, the virus which is very closely related to HIV and which affects the monkeys, the chimpanzees, basically those other animals. So there is that evidence. HIV is related to SIVs, to some strains of SIVs. And therefore, the basis of this theory is that people think, or whoever came up with this theory, thought that SIV, perhaps it was transferred to humans as a result of, interactions or intake or killing chimpanzees for food through what we call bush meat hunting. So it may have crossed from these non-human primates into human because of killing chimpanzees for meat through what we call bush meat hunting, which is actually a very common practice also in Africa, especially for those communities which live in near national reserves and so on. So, so during butchering of these chimpanzees, it is thought that perhaps the blood from these primates, from these primates, may have gotten into wounds or cuts of the hunter, and later the SIV it adapted in the body of the hunter, later becoming HIV-1 or HIV-2. So that's basically, so the entry here of the virus is through the cuts or the wounds of the hunter as they butcher these animals. And again, that's also the reason why the theory is called the cut under theory, because the entry of the SIV from these chimpanzees is through the cuts or the wounds of these hunters. And again, there's some a little bit of kind of evidence or some support to these theories. One is that we know that HIV has different strains, right? I mentioned that. And therefore, because once these hunters became infected with SIV, the SIV, it developed into different types of strains, okay? That means that the fact that we have different strains of HIV could have been, it could have been because of these SIV, developing into different strains. Every time a person is infected through the opening of the wounds and also through the cuts. And again, also to add on this is that in Africa, we know that there are so many retroviral zoonoses that are still currently occurring. For example, we know about Ebola, right? Very different types of a disease which is hemorrhagic, and which is actually again also thought to have originated from the wildlife, from the monkeys, or from these non-human, or from the chimpanzees and so on. So the fact that we have zoonosis in Africa, which are actually thought to have originated from animals, is also a support to this theory. And even when you look at the COVID situations that we are having currently, and it is also thought that COVID also came from the wet markets, where these animals are slaughtered, right? That's also, again, also pointing out that perhaps these animals are the primary sources of many pathogens that we know they cause very bad diseases in humans. So I really don't want to overemphasize on this. I hope it was clear. So let me mention the other theory. That's the theory number 4. The contaminated oral polio vaccine theory. And I find this theory very controversial and at the same time also thought-provoking. And it is a theory that had many players or many actors who contributed to the development of this theory. But one of the lead player is a BBC journalist who was called Edward Hooper, and who lived in Africa between 1981 and in 1986. Of course, in addition to being a journalist, he also had other employment opportunities. Like, for example, he was a teacher at some point, and he was also a salesperson and so on, a storekeeper, that is. And so Edward Hooper is the main person in this theory. Is the main person who actually contributed to the development of this theory. So, being a journalist in 1986, while in Kampala, he reported that hundreds of people on the shores of Lake Victoria were dying of a mysterious disease, which was believed to be AIDS. And so he went there in the shores of Lake Victoria and interviewed some people there. And again, what he noticed is that all the people who were infected with this mysterious disease had some common symptoms. For example, they manifested the cough, they had fever, they had ulcers in their mouth. There were sores in many parts of the body, and they also had constant diarrhea. And in addition to this, all the individuals were also thin and shrunken, what people refer to as the slims. You know, HIV, the slimming, because of the fact that during those days when there were no drugs, one of the typical sign or symptom of HIV was slimming of people. And so now, having mentioned about all those symptoms which are being manifested by the people, then he suggested that perhaps HIV could be traced. He suggested that HIV could be traced to a vaccine which was being administered in the Central Africa, in countries like Congo, Rwanda, Burundi, in the 1950s. And the name of this oral polio vaccine was called CHAT, which of course had been administered to close to 1 million people in those countries which I have mentioned. And so because in the development of this vaccine, the people who came up with this vaccine actually used, they grew the virus using kidney cells obtained from local chimpanzees. So he thought that that the CHAT vaccine, that the CHAT oral vaccine was contaminated with this HIV virus from these chimpanzees, because of the fact that in the development of this CHAT vaccine, they had used kidney cells that were obtained from the local chimps. So he claimed that people who were vaccinated using this contaminated vaccine with HIV virus later became infected with HIV. And again, there was also a lot of, there was a study again, which again also pointed out that in areas where this vaccination was administered, there was actually higher cases of HIV. So I don't know whether it is true, because it's a controversial theory, but again, it is something that perhaps we can also interrogate later. And there's also the theory number 5, the colonization theory. And again, this theory is like a combination of the cut under and the contaminated needle theory. So it is based on the premise of these two theories, and it was proposed in 2000 by an American specialist in primate behavior who was called Jim Moore. So this guy, he published his work he published his findings in the Journal of AIDS Research and Human Retroviruses. So he's the main person who came up with this colonization theory. So let's look at what does it state. And we all know that in 19th century and also early in 20th century, many parts of Africa were actually under the colonial rule, right? For example, in Equatorial Africa, most of these parts were under French, and again in Congo, again these people were under the colonial rule of the Belgians, who were actually very harsh in terms of how they treated people. And so during this colonial period, these guys, they created the labor camps. They created labor camps where people were forced into labor, again under very poor sanitation conditions, where again also food was very scarce, and there was a lot of demands in terms of in terms of the physical activities that people were engaged in. So because of all those conditions, poor sanitation, lack of food, being scarce, and of course engaging in physical labor, that Jim Moore postulates that perhaps this could have created poor health among the people who were infected, and that could also, it could have created an enabling environment for the HIV transmission to take place, and later perhaps the HIV transforming to become the HIV. So the poor conditions created poor health outcomes among the people, and of course that also weakened their immune system. And all that combined, all these factors combined, could have created an enabling environment for the transmission of HIV, which is not perhaps transformed into HIV/AIDS. Now, further to this, these individuals who are engaged in forced labor, they also used to feed on stray and also perhaps sick chimpanzees that were potentially infected with HIV. And again, that is also thought to have led to the transformation of the HIV into HIV-1 and also 2. And again, also to add on this, Jim Moore also believes that these individuals who are forced in labor could also have been vaccinated with unsterilized needles for diseases like smallpox, for them to remain alive and to keep on working. So again, when you think about that, because I said that this is a theory which actually puts together the cut under theory and the contamination needle theory, then when you think about that, perhaps also this HIV transmission were enhanced through these unsterilized needles, which were being used to inject or to vaccinate laborers against some diseases like smallpox, to enable them to remain alive and to keep on working for these colonial masters. And one interesting aspect, again, of this theory is that these guys, these colonial masters, also encouraged, but they also actively employed sex workers to keep the workers happy. Okay? So again, that also creates another possibility for transmission of HIV, because that could lead to multiple sexual partners, and so on and so on. So if one sex worker is infected, that could lead to multiple infections within the same labor camp. So the support for this theory is the fact that HIV was first discovered or was in circulation during the same time, that's in the 20th century, when again these forced labor were happening. So that's the only support for this theory. The fact that the HIV infections were high at the same time when these forced labors by the colonial masters were also prevalent. So that connection between the two is what it is actually what supports this theory by Jim Moore. We still have some few minutes. We are supposed to end our class by 9, and so I will not take much time with the remaining 2 or 3 theories. I'll just mention the key message for each, and so that we don't spend much time beyond the 9 when we are supposed to end our class. So there's also the conspiracy theory or beliefs, I would say, and I know even when we talk about COVID, for example, maybe to start from there, we also have conspiracy theories about the origin of COVID, right? Where some people think that COVID was perhaps manufactured by the China government to kind of control the population of other people, or even the involvement of some billionaires in the creation of this virus to cut down on the population of some continents like Africa and so on, or even to create a virus so that it disrupts economies of other countries, and to ensure that China is able to sell drugs, vaccines, and other ingredients that may be required in the formation of this vaccine. So conspiracy theories are not new. That's what I want to emphasize. They're not new. They've been there, and we are still grappling with them with respect to some of the recent pandemics that, like COVID, that we are experiencing. So what does conspiracy theory explain? So they usually try to attempt or to explain an harmful or a tragic event as a result of the actions of a small, powerful group. They try to explain harmful or tragic events as a result of actions of a small, powerful group. That's the example that I gave about the COVID and how some people might think that it was manufactured to control human population in some continents and so on. So when you look at literature, there's a lot of conspiracy beliefs about the origin of HIV and the role of government in the AIDS epidemic. And this is quite common in some populations, like among the African Americans, among the Latin populations, and again, it is less common among the whites and Asians. So briefly, let's look at some of these conspiracy beliefs among the African American populations. What do they say about this HIV? And one thing is that there's a study which was done in USA by someone called Ross and some other scientists, and this was done in 2006. The title of that article is "Conspiracy Beliefs About the Origin of HIV/AIDS in 4 Racial or Ethnic Groups," and there's a link to that article. And I will also be able to post the same in the model. So in this study, they found that about 27% of the African Americans actually believe that HIV/AIDS is man-made. It was created by their government to kill and to wipe out the black people. So that is an example of a conspiracy belief about the origin of HIV. And further to this, the study also found that 20% of the black American men strongly agreed that AIDS was produced in the government lab to control the black population. And again, also in the same study, it also highlights that more than 50% of the African Americans also believed that there is a HIV vaccine which was secretly being withheld by the government from the poor. So it is only accessible to the rich and not to the poor. So the main challenge with these conspiracy beliefs is that they kind of hinder the implementation of control interventions for HIV. For example, conspiracy beliefs usually they are significantly associated with negative attitudes towards condom use or even inconsistent use of condoms among the Black people. So they hinder the implementation of control interventions against HIV, and they are not true. We should always counteract them with the facts that HIV, based on the evidence that we have, it was not manufactured in the lab, and there's a lot of evidence which points that perhaps it originated from the animals, just like the case we have with many retroviral zoonoses that I mentioned in my earlier slides. And there's also the number 7 is the monkey origin theory. It's a recent one, and this is based on a study which was done in 2010 by someone called Paul Sharp, someone from the university, from Nottingham University, and also Beatrice H. Hahn from University of Alabama. And from this study, there's actually a compelling evidence showing that HIV originated from the chimpanzees based on the, I don't want to say sequences, because that would be quite complex for you, but based on the data which they found from this study, there's actually compelling evidence that HIV probably, or most likely, it originated from the chimpanzees, and the virus, perhaps it may have crossed through, several species before it got into humans. And one of these species, perhaps the gorillas and so on. So again, I want to invite you to look at this study. You can look at the summary of the findings from this study, which points out HIV-1 could have originated from the monkeys. And I think now the last one is the mysterious origin theory, which I find it quite unfounded. It is not that strong, very weak. And this theory, they suggest that that viral material was carried by a comet passing close to the Earth, and this material was deposited subsequently infected nearby people. I want to invite you to also interrogate this theory. In my own view, in my own opinion, I find it also very weak, and it is not that convincing. So that is the end of my lecture. I think we still have some few minutes. I can check the questions if there is any that I can answer. In terms of this content for this lecture, I will upload it straight in the module. Of course, there were some other materials which I had shared with you to prepare for the lecture, but the PowerPoint, I will be able to upload it in the module after this class. And again, I will also be able to provide a link to the class for those who may have missed one or two things and they want to understand what I was explaining. So let me look at the comments. And what are you saying? There's another theory which I will not be able to mention, that is calculation theory. Please, it is in the notes. And I think that can be part of your assignment for today. Look at it and try to write like a half-page summary about that theory. Theory ya mwisho hapana. Yes, I think some of you also have some comments about this theory. Some of you are not very much convinced with the last theory, and so on. So I see we have a few of my colleagues who are online. I really don't know if you want to say one or two things before we wind up the class. And before they say what they want to say, and let me mention that after this class, you're going to meet your lecturers in your respective venues where you met last week. And if you met your lecturer on Monday or Tuesday, or Thursday or Friday, it means that you'll have to wait up to next week to meet them. Okay, is that clear? If you met them on Wednesday, then from this class you proceed to your venue and wait for them there. This is very important because we need to get a class list. You know, we need to have you sign the class attendance, and also to seek for some clarifications, because the comments are too many, and I may not be able to answer all these questions given the limited time that we have. So I don't know, Dr. Franklin Nambuga is logged in. I don't know if you want to say anything, Dr. Michael Kitaiga and Dr. Julius. Do you have anything that perhaps you want to say before we wind up the class? No, I don't see any comments. So please, let's say meet in our respective venues, and I wish you a very good day. Bye.